Welcome, everybody. My name is Derrick Rossi. I'm the interim CEO of the New York Stem Cell Foundation. It is my pleasure to welcome you all today to this webinar in which we will discuss how stem cells, and I guess more accurately, how cells derived from stem cells are being used to treat age-related macular degeneration, or AMD for short. First, a few basic facts. For those over the age of 75, the risk of contracting AMD is nearly 1 in 3. As of today, there are over 11 million Americans affected with AMD, and by 2025, it is estimated that 22 million Americans will be affected with AMD. So this is a massive clinical problem and unmet need. I think really importantly, from the perspective of developing disease-modifying therapies, AMD is a rare disease in that we know exactly what type of cell is affected. It's actually only one type of cell that's damaged and needs to be replaced. And this is cell type is the so-called retinal pigment epithelial cells, which we'll hear a lot about today. Short for that is RPE cells, which sit in the back of the eye. And at NYSCF, we've been working on this for several years. We saw this as an ideal opportunity to treat disease with stem cell-derived cell replacement therapy, because we can create essentially healthy versions of damaged RPE cells from a patient's own stem cells that could then be transplanted back into the affected person via autologous transplantation, meaning the cells derived from the patient itself, which of course has the really great advantage of not eliciting a rejection of the transplanted cells, because they are in essence the patient's own cells. And so as of today, we've made great progress towards establishing this cell-based therapy for AMD. We've teamed up with a group of really outstanding internationally recognized scientists and clinicians who are leaders in the field, including Dr. Stanley Chang and Kapil Bharti, who we will hear from today, and whom Dr. Raeka Aiyar will introduce shortly. We have established and optimized the techniques and methodologies for creating first stem cells derived from patients, and then secondarily for converting them into these RPE cells. And this has really relied on adopting Kapil's technique for doing so, which we'll hear more about. I think really importantly, we've built a GMP suite, or this is the so-called clean room that one needs to produce clinical grade cells in for autologous transplantation. So we have a GMP suite at NYSCF, and we've designated its use for making RPE cells for autologous transplantation. And I think most importantly, we have a fantastic team here at NYSCF and are very proud of the progress that we've made towards pushing this clinical trial closer to patients suffering from age-related macular degeneration. And this will be NYSCF's first cell-based clinical trial that we've run, and I fully expect that we'll take the learnings from this therapeutic avenue from this and apply it to other debilitating diseases. So we will learn a lot from this, which will usher in future therapies for other diseases. Really, that's all I wanted to say. So I hope you enjoyed today's discussion, and I'd like to introduce you to today's moderator, NYSCF's Vice President of Scientific Outreach, Raeka Aiyar. Raeka, please.
Thank you, Derrick. I'm honored to be here and to do with this amazing team and to moderate this exciting discussion about our first cell therapy program here at NYSCF, focused on age-related macular degeneration. Without further ado, I'd like to bring our panelists in and introduce our first panelist, Stanley Chang, who is the KKC and Kuteying Professor of Ophthalmology at Columbia University, an ophthalmology specialist who's pioneered many surgical techniques for AMD and related diseases, and will be a surgeon in our forthcoming clinical trial. So Stanley, thank you so much for being with us today. As a physician who treats AMD, can you tell us a bit more about what exactly this disease is and what the patient experience of it is?
Well, AMD is really the leading cause of legal blindness in our senior population, and patients lose the ability to see detail. They have difficulty reading, and in the more severe cases, they have some more difficulty ambulating. So this is a very major problem. We feel that, as Dr. Derrick said, that it has a high incidence, as high as 8 or 10% of the population will lose vision from AMD as they become older.
Yeah, certainly a significant burden. And so thank you for being with us, and we'll come back to the treatment options later on, but I want to first bring on our next panelist, Kapil Bharti, who is a senior investigator in the Ocular and Stem Cell Translational Research section at the National Eye Institute as part of the National Institutes of Health. Thank you so much for being with us, Kapil. It's really great to have you. You are a leading researcher in the AMD field, and as Derrick mentioned, there is kind of this one specific cell type that's perturbed and sort of damaged in the disease. Can you tell us what's known about the causes of AMD and what are these cells and what happens to them in the disease exactly?
So Raeka, thank you. Thank you for having me here. I'm really excited to participate with this excellent panelist here. So AMD, as Stanley talked about, is a debilitating condition. Of course, the vision loss, we all understand, in AMD happens because patients lose the photoreceptor cells. So these are the cells that perceive light and send signals to the brain. These are the cells that allow us to see. But these cells die in AMD patients because there's a layer of cells present adjacent to photoreceptors called the retinal pigment epithelium, in short, the RPE cells. These are the RPE cells that die off and degenerate as the patients get progress towards later stages of AMD. And you can see these cells behind, I guess, in my background, each cell is a very high magnification, and in Stanley's and Cecile's background, you can see them as a smaller green hexagons, if you will, and they're arranged in a perfect hexagonal morphology as a honeycomb structure. And with time, these cells just peter out and die, and these cells have been supporting photoreceptors' health and functioning throughout their life. And as RP cells die, photoreceptor cells die off, and that's why patients go blind. The causes that we know of for AMD, of course, one of the biggest risk factor is aging. Two additional risk factors that have been identified is diet and smoking. Of course, it's a polygenic disease. What that means is there are many small risk factors throughout our genome that are present that also increase the likelihood of AMD. But really, I think aging, diet, and smoking contribute to that or add on to that and increase the possibility of us progressing towards advanced stages of AMD.
And what would those dietary factors be that are sort of identified as increasing risk?
I think the biggest risk factor in terms of diet is high fat diet, or you can also consider it as a higher BMI ratio of people. So really, diets with high fat, I would put it out there, is one of the biggest risk factor for AMD.
Thank you so much for that explanation. We'll definitely come back to interventions shortly. I want to bring on our next panelist, Cecile Terrenoire, who's one of NYSCF's own scientists, a senior director of process development, and she leads the processes of the development of processes and quality control methods that we're using to make these cells for this therapeutic program. So, Cecile, can you tell us how you use stem cells to make these RPE cells that are damaged? What does it take to make these beautiful cells behind you into functional cells that can actually help a patient to see better?
So thank you, Raeka, for the nice introduction. In order to turn stem cells into those retinal cells, those RPEs, We are using a recipe that was developed by Kapil. It's a really amazing recipe that works very well. It takes about 2.5 months to turn a stem cell into retinal cells. I have a few props that I can use to try to show what we're doing in the lab. So in this tube, I'm going to bring it closer to the camera. At the very bottom of it here, sorry for this, we have a little white mass of cells. These are stem cells. In here you have about 1 million stem cells. We put them in a petri dish. We feed them in a way with a soup that contains all kinds of chemicals, vitamins, growth factors, and chemicals that the cells are going to swallow. They're basically going to drink the soup. And those chemicals will progressively turn the stem cells into retinal cells. And I'm going to show you now those retinal cells. So if you're looking at the bottom of this tube, see that they are pigmented, they're black. Here again, you have about 1 million retinal cells in this tube. This is one of the features, the pigmentation of those cells is one of the features of those cells. And so what we're doing basically in a petri dish, it's to mimic the development that naturally occurs in a human being when the retinal cells are being formed. So we have the capacity to do that thanks to this amazing protocol that Kapil developed. And once we make those cells, after 2.5 months, those cells are going to be basically, we're going to create a little patch of retinal cell. So I'm going to show that again with another prop. And what the patch is here, it's this teeny tiny thing. And I'm putting on the side a pin just so that you have a sense of size. This is really, really small. This is smaller than a Tic Tac, just to give you an idea. But with this patch, we're going to be able to do something really amazing. This is what's going to get transplanted in the back of the eye of the patient to help cure the disease. So it doesn't look like it's a lot of cells, but the potential is really, really amazing out there.
Thank you so much. That's really, really exciting to see. And the scale definitely helps. And I always found it fascinating that the most beautiful cells are the ones that actually help us to see them. It's a little bit meta, but all right. Well, last but not least, I'd like to introduce Howard Kim, who is the Vice President of Cell Therapy here at NYSCF. Howard brings a ton of experience in this field of cell therapies that are created using stem cells, leading collaborative and complex projects like this one, and now leads our cell therapy programs here at NYSCF. So, Howard, I wanted to ask you to start with, why are cell therapies such an exciting avenue in general, not just for AMD, and what does bringing a cell therapy from the lab to the clinic entail?
Great, thank you, Raeka. Cell therapies to me kind of represent the next frontier of medicine. Really the majority of our diseases are ultimately the result of cells that are in our body that are impaired or diseased or just frankly lost or dead. This is not just the case for AMD, but also, things like Parkinson's, the after effects of stroke or heart attack, diabetes, et cetera. And in many cases, current treatments for these diseases, really help manage the disease, whether that's by slowing disease progression or managing its symptoms or, replacing one aspect of what a cell would do, for example, generating insulin in a diabetes model. But by nature, the cell being what's naturally occurring in our body, if you can replace those in the disease state, you're really going for a cure, which is, the ultimate goal is to have something where patient doesn't have to take a drug every week or et cetera. It can be a one-time, hopefully, treatment that reverses all disease aspects and brings them back to sort of a normal state. And then the second part of the question is, what does it take to bring it to the clinic? Cell therapies is in principle similar to any other, drug therapy that has to go through the FDA for approval and then clinical trials before being sort of broadly available. The real kind of challenging aspect to that is that it's a living drug. So, whereas if you had, a chemical, it's very stable, easy to analyze and measure. Cells are living things; they change. They're not as stable. They can't be, sterilized, for lack of a better word. So just the environment of understanding its mechanism of action and the manufacturing and characterization aspect of it is a much bigger challenge.
Absolutely. Thank you. And thank you for pointing out the idea of it being a one-time therapy. So that is the idea with this therapy that you're all working on. That was a question that came in from the audience. And I want to thank those who submitted questions in advance and encourage you to use the Q&A function if you have any that come up as we continue to go along. So let's talk now about this specific cell therapy that this team is working on as a relatively new avenue for treating AMD. I'd like to put it in context. So Stanley, can you tell us a little bit about the current treatment options that are available for AMD patients? And, there are these different forms of AMD, dry versus wet, and sort of what that kind of landscape looks like.
Yes. There are two forms of AMD. They start, early AMD starts as little deposits called drusen in the back of the eye at the layer where the RPE cells reside. And over time, these drusen will increase with age, and then gradually the RPE cells will die off. And this atrophy or death of the RPE cells results in what we call geographic atrophy, which accounts for dry AMD, and about 80% of macular degeneration, 80 to 90% are caused by dry AMD. In the later stages, some 10% will go on to form neovascularization, which is the growth of scar tissues or blood vessels that can further bleed or cause damage to the retina and the photoreceptors. And this is the so-called wet form. We have a pretty well-established treatment for wet macular degeneration, but in the past, there's been no treatment for dry macular degeneration. So these patients continue to lose vision over time. Over a period of maybe 2 to 5 years, they may progress and lose the central vision in the eye without treatment. Most recently, there has been a new drug approved called now called Civofir, which is from the Apellis corporate company, in which there's been an injection of a small molecule that inhibits complement. In these patients, it only slows the progression of the disease, and still a large percentage of patients continue to lose vision over time, and the geographic atrophy worsens. They've shown in the most recent published studies of this drug that about 30% get slowing of the progression of the geographic atrophy, but most of the patients maintain the same visual acuity or get worse over time. So there needs to be a better treatment. There needs to be a treatment that can actually recover vision and regenerate those cells that have been suffered and died from the dry macular degeneration process. The other problem with this treatment is that you have to go to the doctor every month to get the injection. And we may not see the effects of the injection till at least 6 months. The best effects are seen after 6 months of therapy. So it's going to be a long haul for the patient to go every month to the doctor and receive an injection. The complications of injections can result in infections, which can result in severe loss of vision. And also, it requires the patient often to go with somebody else to the doctor every month. They need to be driven there. They can't go there on their own. It's hard to go there on your own. So we need to find a much better treatment than is currently possible.
And what do you think then are the advantages that a stem cell treatment like what you all are working on for dry AMD would bring, that, the gaps that it would fill that patients don't currently have options for?
Well, we hope to, with this treatment, this pioneering treatment that Kapil has started, that we would implant these cells, they would survive and flourish and connect with the overlying retina and function and improve vision. And some preliminary studies with embryonic stem cells have shown that a small group do improve in vision after a year of transplantation of embryonic stem cells.
Right. This was with Pete Coffey's work in the United Kingdom years ago. Yeah, this
Yes.
was something that NYSCF supported years ago, which was sort of the genesis of this current program, and certainly promising to see what came out of that. And we're hoping to build on it here. But the contrast here, those were using embryonic stem cells, which are just, sort of a single cell source. Here we're using induced pluripotent stem cells that are derived from the patients themselves. Can you talk about why that's important?
Well, the immune system, you often, if you get a transplant of somebody else's cells, the immune system comes into play, and you often get rejection. So you have to take medication to stop the rejection of foreign cells that are implanted in your body. With the iPS stem cells, they are from your own body, your own tissue, and presumably the issues with rejection are not going to be a problem. Kapil has done a lot of work with this. He could explain this further, I suppose.
Yeah, please feel free to add, Kapil.
I think the only few things I would want to add on to this is it's really, I want to emphasize that we are hoping this is a one and done treatment. This is not, these patients are not going to have to go back over and over again. And we are replacing the cell type that we all talk about, it's present in our back. We're replacing the degenerative tissue. This is not a drug per se, even though it's alive, we can call it a live drug, which will interact with cells, but it is replacement tissue. It's a regenerative therapy. And as Stanley pointed out, this is made with patients' own cells. So we expect that in the long term, or many years, we're talking 5, 10, 20 years of patients' life, the cells should continue to stay stable and graft better. And patients should not need to take any immunosuppression or any drugs to keep the cells alive.
And this is an advantage because the immunosuppression therapy can be detrimental in other ways, correct?
Exactly. Yeah.
Older patients don't tolerate immunosuppression very well.
And what would be the disadvantages of having to take immunosuppressive therapy on top of this, Stanley?
Well, immunosuppression increases your risk of other infections. For instance, if you are immunosuppressed and you could get COVID more easily or other serious infections.
Yeah.
There may be also an increased rate of secondary cancers with immunosuppression.
So definitely a lot of good reasons to avoid that. So as we talk now about this specific program, which using the patient's own cells, otherwise known as autologous type of therapy, Kapil, you've recently launched a clinical trial at the National Eye Institute, and congratulations on getting it to your first patient a few months ago. We're all really eager to hear, and many questions have come in, about what the latest status of this is. So can you tell us a bit about what this has entailed so far, and what more can you tell us about the trial and the progress?
Sure. Thank you, Raeka, for your wishes. Yes, the trial was a major milestone for us. Coincidentally, was approved by the FDA right before the COVID pandemic started, and we had to shut it down because of the pandemic. But we were able to restart the trial last year and enroll a few patients and transplanted the first patient in actually August last year. We're in the process of enrolling additional patients. So currently, since this is a phase 1/2A trial, it's still considered a safety and a feasibility study with a primary endpoint of 1 year and secondary endpoint till 5 years. We'll follow up these patients. But at the 1 year mark, we could talk about the patient data and how the patients are doing. I think that the important point to remember is that at this stage, the trial is active. Patients who are interested in enrolling in this trial, they should reach out to NEI. We have on our website, the information for the trial is out there. You can also go to clinicaltrials.gov, and you will find the links to our patient coordinators and their contacts so that you can reach out to them and participate in the trial. One important part for us was while we were working on towards the trial, and Cecile nicely illustrated the process, how we treat the cells with different type of soups, and we have recipes to transform the stem cells, iPS cells into RP cells. It was very important for us to really understand that the biology of cells and make the right kind of cells and make a process that is going to be robust enough that every single patient, we could make their cells over and over again and make sure that the cells are functional as we transplant them in the eye, because we're transplanting them in older eyes. We want fully mature cells that will hit the ground running. We talk to the overlying photoreceptors, would protect them from dying further. And one of the key parameters we used was, again, the morphology of cells. These cells, as I pointed out earlier as well, have perfect hexagonal morphology. They're like honeycomb shaped. And that morphology is attained as the cells become more mature. And as Cecile pointed out, they also become pigmented as they mature. It's the same pigment that is present in our skin. And we realized that we could exploit these two features to actually develop an artificial intelligence-based measurement that could tell us whether the transplant was ready for transplantation or not. So all we have to do is, after we trained our machine learning algorithm, all we have to do is take a picture of the cells as they're being manufactured, and then the program tells us whether they're ready for transplantation. Very simple assay. In fact, one of the first uses of AI in quality control of cells that are going to be transplanted in patient, and telling us that the cells are fully mature, fully functional, and they will do their job as soon as they're transplanted. They won't even have to wait a day to start doing their job.
That is really exciting, getting AI to see things that we can't see. Okay, I'll stop with the vision puns, but Howard, I wanted to ask, sort of, you're leading NYSCF's efforts to launch a parallel trial to NEIs. Can you talk about why this is an important complement to the trial? And Kapil too, please chime in. How are these both of these efforts going to advance the field?
Great. Yeah, so what NYSCF is doing is we've licensed the technology from Kapil. So we're essentially trying to launch a sister trial where we're generating the same drug product, the same patch as Kapil's, with specific changes based on NYSCF's facilities. But really, what the aim is to broaden access to this exciting novel treatment. So, the more people working on this, the more data that'll be generated, the more patients can be provided this type of treatment, both in the short term and the long term. And I think that's really the goal here.
If I may add a comment.
Please.
So one of the things that's really important in those trials is that once the cells are made in a clinical lab, like the one we have at NYSCF, the idea is that the hospital where the transplant is going to happen has to be within a few hours of driving by car distance. It cannot be we cannot ship the cells across the country. So that's why the manufacturing site needs to be really nearby the hospital. And so with the high demand, the large number of patients in the country, everywhere in the country, you need to have those sort of like satellite places where there's going to be manufacturing and transplant, in the same area, in the same geography.
Perhaps, Howard, you could describe what the process is, because it's a very rigorous, very precise project to grow iPS stem cells into RPE cells. Can you describe how much blood is required of the patient and how much time it takes, so the audience can understand how careful this process has to be and how very tightly regulated the FDA requires?
Yeah, of course. Yeah. So very broadly speaking, it starts from collecting whole blood from the patient. So that's a blood draw. So the NYSCF process and both in NEI's process start with blood. And then it's roughly a 3-month process to generate that patient specific stem cells and to test them. So there's a battery of tests to ensure that they're genetically stable, that they're sterile, et cetera, and then the process to go from stem cells into the final mature retinal pigmented epithelium is about another 3 months as well. So we're looking at sort of at least a minimum of 6 months from patient to treatment in this model. And then all that, as we'll probably allude to later, has to be done in a very controlled environment, clean room, highly trained staff, very strict regulations on equipment maintenance and reagent selection, et cetera. So yeah, it's quite a big effort to get this piece there.
Yeah, robots nourishing these cells day in and day out, 24/7,
Yeah,
making sure
absolutely.
they're properly cared for.
Exactly,
and the other
replicating
thing-
that environment in the body. Yeah.
The other thing
Cecile.
we could add is that, I mentioned millions of retinal cells in that tube, and then, I think it's less than 100,000 in that little patch that I showed. We typically manufacture millions of cells to generate one little patch, but it's mainly because the cells that are not used to make the patch are used to for quality control, to double check that the cells we're making are indeed retinal cells. There's an array of tests that we're using along the process to make sure that everything is working as it should, not just in terms of making the right cell type, but also making sure that we don't have any contamination, there's no bacteria, viruses that could potentially be, a problem down the road. So most of the cells we're making are actually for safety purpose, to make sure that everything is fine, to demonstrate that our little patch is going to be fine and that it's going to work as it should in the patient's eye.
And we'll be safe. Yeah. Kapil, what did you want to add?
I just wanted to add on to what the other panel members have already said, is that I think the key point about the rigor is the safety here and ensuring that not a single iPS cell, not a single stem cell is left in the final tissue that we are transplanting. I want the audience to really fully understand that we are transplanting a small tissue of eye cells. There's not going to be a single stem cells when the transplant is ready, and using multiple techniques, as Cecile pointed out, and using the AI technique that we have developed, we ensure that we look at every single cell before it's going in the eye to make sure that patients are going to be safe. So I want patients to understand this difference between the snake oil, the Hook stem cell therapies that are out there versus these kind of legitimate treatments that have gotten FDA approval, where we are going at length to demonstrate safety of it. So just wanted to make sure the audience understands the difference.
Thank you for drawing that distinction. Certainly having stem cells be transplanted would be problematic in a lot of ways because they will keep growing, and that's, absolutely not what we want. And that's why all of these different safety checks are really, really important. I think that's a good transition into talking about sort of the regulatory path that surrounds these therapies. This is all, a relatively new type of therapy. And so there's obviously a lot of unknowns around it. So what are the steps that need to be taken to make sure that these therapies, that these particular cells that you're making are going to be as safe as possible? You talked about some of them with, avoiding contaminations, making sure that there's a pure cell product of retinal cells only and no stem cells left. Are there other things that are required by the FDA as we advance towards a trial and through a trial? Howard, do you want to, or Kapil, why don't you start? Yeah.
I think, two things we already mentioned. One is ensuring that there's no contaminants that can cause a harm in terms of any infections to the patients. And the second is contaminating cells, be it iPS cells or any other non-RP cell type. So we ensure that these are pure RP cells that are matured on a small scaffold surface before they're transplanted. Of course, the biocompatibility of the materials that are used as xenofreeness, so there's no animal component going into patients, and the surgical tool and techniques that are used, they are all they're not going to cause undue harm to the patient. So those are the few things that were required by the FDA before the product or the trial was approved. I don't know, Howard, you want to add anything else here?
Yeah, I think just from a very broad level, the FDA wants to know that you can manufacture it robustly, that you know what your product is, and you can you have tests that can measure it, whether that's things like sterility, but also, as Kapil mentioned, functionality of the cells. And then in general, there's an aspect of what your therapeutic target and application is. Do you have an idea of what the mechanism of action is, and do you have any proof of principle? Really what the FDA wanted for you to start your phase 1 trial is positive benefit over risk argument.
And that's how you determine who is eligible for a phase 1 trial. There have been some questions about that coming in. Who is eligible for this? How do you sort of determine in terms of age and the stage of the disease?
I'll start, and I think Kapil or Stanley can add on to this. But in general, phase 1 trials are aimed at safety primarily. So a lot of times you are looking at the more advanced disease population. I believe in our trial, both Kapil and ours, we're planning on say for vision range 2 over 100 or worse, and that's really to show that if you're, we want to show it's safe and the foremost, and the best way to do that is with patients who are sort of later in the disease stage where you can't do as much harm potentially for someone who has relatively good vision at the moment.
Stanley, Kapil, anything you would like to add to
Probably
that?
start with patients who have the worst eye of a patient, and where the disease is quite advanced, to make sure that we can implant the scaffold of cells and see it integrate, because the surgery is very precise and surgery, and very important to have the surgery done properly. So we have to make sure that every factor is working well before and then we have a very careful way of following the patients, see how well this tissue integrates into improving the visual function. How do we assess the function of the patient? We have very precise imaging and other technologies to look at the functional aspect of this transplant after implantation.
So what is it that these cells are doing? How will we know that that they're actually working? Is it through the restoration of vision? When do you expect that to actually
We would
kick
look
in?
at where the focus of vision is, and whether the implant is actually providing signals to the patient that they can see over the implant.
Okay, and there was also a question about the recuperation. Once a patient receives the transplant, how long does that take? What does the sort of recovery look like?
Well, we would hope that over time there would be further improvement. We don't know exactly how long the vision would take to improve, but we would like a follow-up of at least a year to know whether the vision's going to improve in the patient. Certainly within a few months, we would have some idea of how well the surgery went, but there may be continued improvement over time. These healthy cells may also rejuvenate some of the surrounding cells that are borderline in function, that they're starting to get worse. So having some healthy cells around them may help reinforce the survival of the cells that are aging around the implant. So we're hoping that will also provide some benefit to the patient.
Absolutely. There was actually, that's related to a question that came in around the photoreceptors, which you'd mentioned earlier as also being, damaged. And is there a chance that by only transplanting the RPE cells that maybe you're not quite restoring photoreceptor function, or has that been shown to be overcome with this approach?
We would hope to select patients that have some degree of photoreceptors still remaining. We expect that those would be the best candidates to get some recovery of vision.
Yeah. And would there be scope, do you see potentially going forward with this kind of therapy that, you might integrate photoreceptors, or it might be available to patients at, sort of different stages?
So phase 1 would be the safety, to make sure that we don't cause anyone to lose vision as a result of the procedure. And then the phase 2 would be to look at the efficacy on a wider scale, larger group of patients, to look at the functional outcomes and the ability of this process to recover vision.
Got it. Okay. I mean, this is I want to talk about this team a little bit, because this is, a really a lot of diverse expertise and a lot of groundwork that has gone into making this, making all of this possible. Can you give us a little bit of insight into how this collaboration works? What does it involve? How do you all work together to make this happen?
A lot of communication.
Which we were hoping would be more done in person, but we had to
Yeah.
be more creative during the pandemic. So when we did the technology transfer to the NYSCF side, we were hoping that the teams would go back and forth and learn from each other's expertise, but we had to make videos of key activities that were to be done as part of the protocol and transfer to Cecile and Howard, and they're already good enough that they knew that what to do looking at the video, and they would make their own video of what they learned from our technology, send it back to us, we'll review it. And in fact, that is still continuing as of today. Even this week, we have been exchanging some videos of some of the key aspects of the process as the NYSCF team keeps advancing into further stages of the IND process to submit their paperwork to the FDA very soon. So I think, as Stanley pointed out, communication is the most important thing. It became challenging, but I think because of the devotion of teams on both sides, we managed to get it through.
Cecile, anything you'd like to add, being on the receiving end of some of these video protocols and all of this?
It's been a truly amazing and wonderful collaboration. In the past I collaborated with other institutes, and I think this is the first time that I have collaboration that works so well. And as Kapil mentioned, this really requires everybody on both ends to truly understand how to communicate with each other. Part of it is like how often you communicate with each other, but also how people need to understand, that the quality of the communication is really important. And we've all been on the same page. It's been really amazing, really wonderful. We're very thankful to Kapil's team.
As surgeons, we're really motivated to help our patients because we see the frustration they go through. If we can really make this happen and work properly, it's going to be a game changer for patients with macular degeneration.
Well, we really hope so. Absolutely. Howard, we've had a lot of questions, of course, coming in as well about the current status of NYSCF's programs. We talked about, all of the work that we've done to advance towards to clinical trials. What can you tell us about the latest? One of the pieces was, of course, building this GMP suite that several have mentioned. Maybe you can talk about the latest with that.
Yeah. And when we embarked on this a few years ago, we were really starting from scratch here. So a lot of the initial, early years were really around infrastructure building. And a lot of that, takes time, money, and people, which, we're of course very thankful for NYSCF supporters to be able to do this. So, we've built our GMP facility, our pharmaceutical grade clean room. We have the infrastructure around all of that, and how to monitor the room, and then on the process side, again, working with Kapil and his team to bring in their process. It's not as simple as being able to do it, but also, formally having that technology transfer documented and performed, several times, which we've done at this point. And then on the regulatory side, interactions with the FDA, we had our what they call a pre-IND meeting, which is your kind of meeting where you present a lot of your preliminary data and they give you the feedback on the last steps that you need to go through. And so we're really just going through those final checks and balances for the FDA before we put in that submittal.
I'd like to give credit to Susan Solomon, who was the past president of NYSCF, who had the foresight to really initiate this collaboration and bring us all together. So unfortunately, Susan is not going to see the fruits of all her hard work, but we really should give her all the credit for bringing us together.
Absolutely. I think she's watching in her own way and would be really happy to see how quickly this has been advancing, even despite the pandemic delays. So
if I can add one little comment.
Please.
It's also obviously thanks to Susan that we were able to build this clinical lab, what we call the GMP suite. She found a very generous donor. And so the philanthropy has been really, really helpful in helping us develop this program. I don't think we would have done this as fast as we have without this effort. So we're really thankful.
Yeah, thank you for raising that, because the GMP suite is also something quite unique. We don't have so many of these in New York City, or kind of in general, as the being the only place where it's really allowed to create these kinds of cell therapies. Can you talk a little bit about Howard and Kapil? Maybe you can weigh in on the importance of having this kind of facility and the need for that as the cell therapy field grows.
Yeah. So. Sort of in lay terms, it's a pharmaceutical grade clean room where all the activity happens, but it's not just the room itself, it's the people, it's the training, it's the what we call the quality system around that, all the protocols and review processes, and maintenance of equipment and materials, et cetera. So it's a lot of infrastructure to build and maintain. The cell therapy community, it's sort of a growing field, this type of capacity of having sites where this activity can happen is few and far between, and the places that do have it set up are very high demand and low capacity. So so for NYSCF, it was important for us to build this capability ourselves, because we have strong aspirations to invest in the cell therapy field, with dry AMD being our priority right now.
Absolutely. Kapil, anything you'd like to add there?
Not much. I think the important part I think everyone has emphasized a lot is that these are new ways of treating patients and actually stopping disease from really going to advanced stages, and the whole field is going to change as iPSC derived therapies become a common practice. So such GMP rooms and GMP practices is going to have to become a common practice. Yeah. And again, two things I want to add is a shout out to all the people who work inside clean rooms, because it's really challenging working in those so-called bunny suits where you cover from top to bottom. And it's a stressful environment, and the pressure is high in there. So it's shout out to those people. And the second thing, kind of an extension of this for future, is that we're going to have to try and robotize any parts of this. We added AI to a very small part of it, but we're going to have to try to make the manufacturing process more and more objective and automated. And I think this is where, again, NYSCF and their expertise will be key, because that's where they are the leaders in this iPSC manufacturing space, again, because of the vision of Susan Solomon. So I think, again, going back to why this collaboration is so important, and taking this technology to the next step.
Yeah, thank you. Thank you for raising that. I mean, certainly the robotics is something that, NYSCF has invested heavily in building out, and also thanks to the vision that Susan had of being able to scale this up and really create therapies that work for many different patients at a time, create stem cells from many different patients. And we're certainly hopeful that those can be integrated into these types of therapies. Also with regards to cell therapies in general, we received some great news this morning from one of our NYSCF innovators, Malin Parmar, who is working on a stem cell therapy for Parkinson's disease over in Sweden, and they've also gotten into their first patient in the last couple of weeks using this approach. And so it's really a growing field. It's such an exciting time to be in it. And I think we're lucky to have a team like this working towards it. So thank you for all that you're doing. Kind of looking forward, beyond just AMD, we were talking about other ways to restore vision, other diseases like wet AMD. Can you comment on what kinds of lessons or resources we have here that will inform the way that we treat diseases like wet AMD and other forms of blindness, and if there's any other developments in that area that our audience might be interested to hear about?
So I think wet AMD definitely is a relatively easy target, and we already have precedents in the field for that. The work done in London, where they transplanted 2 patients again, we mentioned that before, Pete Coffey's work, with both had an acute form of wet AMD, and the one and the only patient transplanted with another autologous iPS-RP patch was done in Japan, and that was a chronic wet AMD patient. So in both cases, we know that the patients have been stable now for years to come. So definitely this patch has the potential to also work as a treatment for wet AMD. And I would say go even beyond that, even for monogenic RPE-related diseases like Best disease, I think is a classical example, or I would say Stargardt disease, where RPE cells are affected. This tissue can provide a potential treatment option. Of course, for monogenic diseases, we do need to do genetic correction, especially for dominant diseases. It will need to be correct the mutation. For recessive, we can just do gene augmentation and transplant the augmented tissue back in the eyes. So definitely, this is I like to call it a platform technology. We go beyond AMD as we have more proof of concept of safety, but also build upon it to make photoreceptor and RPE dual patch and RPE choriocapillaries patch, so that we can treat many different forms of retinal degenerations and even injuries that affect the back of the eye. One of the projects we are working on in collaboration with David Gam and funded by Department of Defense is we're trying to create the entire back of the eye for war injuries where soldiers lose a part of the eye or part of the retina along with the RPE to really replace the entire three-dimensional tissue. So I think this kind of technology can go way beyond what we're currently using it for.
That's really exciting to hear. I do want to address the many questions that are coming in and sort of, are there ways that people in the audience can sign up for these trials? What might be the timeline for reaching the general public? Of course, obviously, we have to go through these multiple trial phases that you mentioned, but Kapil, Howard, you could comment on, what that timeline looks like, what those opportunities look like for patients to sign up.
So like you said earlier, our trial is active and patients can either go to clinicaltrials.gov, search for it with AMD, IPS cells, and NEI, and they should read the right link. They can also go to nih.nei.gov directly, and we have a link to sign up for this trial. And if they're eligible, they should be able to participate in the trial. And for currently, it's a phase 1 safety and feasibility study. We hope to complete that in the next couple of years and move on to a phase 2B study, which would be more looking at the efficacy of the transplant. Howard, you can comment on the timeline of your trial.
Yeah, and our trial, we're anticipating mid next year to launch it. So we'll have more information available when that time comes.
Once we are able to start recruiting. Yeah, absolutely. And what do we think the timeline is that, what might you foresee as the timeline for reaching the general public for this therapy, these types of therapies to be more widely available?
I think in the next 5 to 6 years, I'll see that these technologies are at a place where FDA is getting comfortable in making them a common practice for people to use it. And it all depends on how successful our initial trials are, our phase 1 trials are. If they show good signs of integration of the patch and we're able to design a phase 2B study where we can directly look at the efficacy of the patch in a larger population and across multiple centers. And again, emphasizing such collaborations across multiple clinical centers, NIH and now Columbia Medical Center, where we can test patch and work with multiple clinicians, multiple surgeons. This is what we need to move on to the next phases of these kind of studies. And hopefully the next 5 to 6 years, we'll start to see significant progress in this space.
What I've done is I've screened some patients for the NEI trial. So patients have come to us interested, and we've taken their images and sent them down to NEI to see if they would qualify for their study. So that's one way that patients won't have to travel from New York before they go to NEI for evaluation. They're evaluated after they're pre-screened by us.
This is an absolutely well-oiled machine. We're almost at the end of our time together. So to close, looking ahead at the likely declining health of our eyes as we're, kind of hunched over computers and devices all day long, and our aging population with diseases like AMD rising in prevalence, is it realistic to envision a lasting cure for AMD and other vision disorders within the next decade? And what makes you optimistic that we might be able to do so? Stanley, I'll start with you.
Certainly, we can improve the quality of vision in some patients, but there are going to be some patients where the disease is so advanced we couldn't do much. So in these patients, we have a lot of work to do. But definitely, if we can develop this technology, we can save a lot of people from being severely and profoundly blind and and keep them functioning, which is really our goal. And I think even in the advent of the wet AMD treatment, we see a lot of patients who the disease has stopped and from progressing, and they can still function to a reasonable level, even though they may have some loss of vision. And that's our goal, to keep them working, functioning, reading, driving, doing their daily work, and giving them a decent quality of life.
And I think to your question, Raeka, as Kapil alluded to, what we're doing here is sort of, laying the groundwork for potential future therapies where that can be, more complex, that can include, say, photoreceptors or other cell types of the retina, so that, the patch that we implant is even more functional and has more capacity to restore vision.
Right, to be able to not just prevent further loss, but
Right.
to actually reverse the loss that's already taken
And
place.
this technology will be useful for glaucoma and other, maybe even optic neuritis. Some of these, there's a lot of work being done in stem cell therapy now, and there's great potential, I think, if we can make these cells integrate into the eye and regenerate tissue.
Cecile, what are your thoughts?
Yeah, I was actually even thinking beyond the eye is that we, at NYSCF, we have great knowledge in how to turn stem cells into other cell types, such as pancreatic cells or heart cells, and so I think the work we're doing right now with this program in making clinical grade stem cells, that's going to speed things up whenever we decide to address another disease, another condition, or using another cell type. We'll be well placed to do that because we have the know-how in-house, and we have the automation, which is also going to be something that's going to be critical to speed things up. So we're really uniquely positioned, I think, to make a lot of progress in these areas.
Thank you. Kapil, I will give you the final word, your thoughts on envisioning cures for these kinds of diseases and cell therapies becoming more widespread.
Thank you, Raeka. So I'll take a step back. I think if we look at it in 2017, really a pioneering work of Dr. Takahashi in Japan transplanted the first patient with an autologous iPSRP patch. And as we all know, unfortunately, they had to stop the trial because of issues with manufacturing. So in less than 5 years, we not only have solved those issues despite the pandemic, we are now doing it at a scale that is much larger, much broader with these collaborations across multiple centers. And I think that is the most important key, that this is now these kind of successes are going to enable so many other groups to work on autologous iPSRP space, iPS cell therapy space. And as Stanley pointed out, for glaucoma and other diseases of the eye, that I think this is going to revolutionize the field, and it's going to increase excitement in further investment in this space. And as Cecile pointed out, through philanthropic funding or through VC funding, there's going to be enough capital put in, enough excitement around this, that there are going to be many more technologies that are going to move forward. I just recently heard a second autologous iPSRP was iPS cell therapy, and this was for a congenital heart disease, was recently approved in the US by a group in Minnesota. So I think we're really at a crossroads, that we're seeing a lot more that's going to come in this space in the next several years. So it's exciting times. I think iPS cells, especially autologous iPS cell therapies, are going to pick up.
Well, we can certainly hope so, and and hopefully this also eases the regulatory path moving forward, and enables these therapies to get to patients even faster, because there's so many opportunities that you all have highlighted for that to really make an impact on patients. So I want to thank you all so much for a really exciting discussion, and for all of the hard work that you're doing individually and as a team to make these therapies a reality for patients who need them. So thank you all so much, and thank you to everyone in the audience who joined us and for your questions. We apologize if we couldn't get to all of them, but we encourage you to stay in touch, join us at a future NYSCF event, and take care.